So today we have the privilege of having Sara McArdle here to talk to us about QuPath. So just very quickly, I'm going to run through sort of a quick introduction. We're going to hear from Sara about QuPath, and then there'll be a Q&A session. We don't have to go all the way up to the hour, but if there's sufficient questions, we have the time. So to introduce Sara, Dr. Sara McArdle is a specialist at the Jolla Institute, Institute of Immunology's microscopy core, where she has a wide range of experience working with different samples and with a focus on, or she's developed a focus on QuPath over the last few years. So I'm happy to, you've all received her bio before, and I'm happy to hand over to Sara. And she was actually so organized because it's very early in the morning for her. We've got a presentation from her, and then she's here for live Q&A. So we can kick off with the presentation.
Hello, thank you for having me here and for giving me a chance to talk about one of my absolute favorite image analysis programs, QuPath. I've been using QuPath since around 2018, and I think it's one of the most powerful, versatile platforms for image analysis I've ever used. So what is QuPath? QuPath is an image viewer and analysis platform. It is free, it is open source, and it works on any operating system, Windows, Mac, Linux, whatever you've got. You could go download it right now at qpath.github.io. It works for bright field or fluorescence images, and it works for stitched whole slide images or single images, if you're just taking one image at a time at a microscope. And it requires pretty minimal computer specifications. It needs a 64-bit processor and at least 4 GB of RAM. Particularly with larger images, the more RAM you have, the faster your whole analysis will go, but for 4 gigs is all you need to get started. And after the program is downloaded, you don't need an internet connection anymore. On the right there is the developer of QuPath, Dr. Pete Bankhead at the University of Edinburgh in Scotland. So whole slide images, these are images of tissue sections or tissue microarrays or blood smears. And they're images composed of hundreds or thousands of fields of view taken individually and then stitched together to form one massive image where you can see the entire slide at once, as well as, and then zoom in and in and in to see individual cells. So why we like to use them is they provide anatomic context. So you're not just looking at cells in isolation. You can see, in this case, this mouse intestine, you can see all of the crypts and see where a cell is in relation to the rest of the tissue. It also gives you a chance to find rare events, maybe only 1 in 1,000 of your cells are the phenotype you care about. And if you're only looking at a really small field of view, you might miss it entirely. And it gives a complete sample overview so that you can tell if some of your tissue is healthy and some is not. It also gives a large number of cells or regions for statistical analysis. I primarily work with tissue sections, so that's what I'm going to be talking about a lot and what all of my examples are. But tissue microarrays or blood smears also work as well. However, these very large images are difficult to work with. They have billions of pixels and millions of cells, so a lot of image analysis programs struggle to really process images of this size. And QuPath is designed with these very large images in mind. And the typical workflow in QuPath is you start with an image, which is composed of pixels, and there can be billions of individual pixels in an image. And you very quickly go from working with pixels to identifying objects. These objects could be your entire tissue, it could be structures like a tumor, regions like an intestinal crypt, or even individual cells or subcellular structures like the nucleus. And then once you've reduced the image down to, say, millions of objects instead of billions of pixels, you can ask questions about the objects, like their size, their shape, how many are there, are they near each other, what's their intensity, all these sorts of questions. I think the best way to talk about the features in QuPath is to actually go through some example projects and show you how I use the program. Frequently, people use QuPath just to look at slides. And it's really convenient because then it can actually open images from most types of microscopes. It has access to the bioformats and open slide library, and it can also work with open source formats like TIFFs. And it also loads image pyramids on the fly, which means it only ever calls the pixels that it actually needs to show you on the screen. And so if you're looking at the entire tissue, it shows you a low resolution image, and as you're zooming in and in on cells, it only loads those exact pixels you need to see at that moment. And this lets it be very fast and lets you look at images that are actually larger than the RAM of your computer. In addition, it has a multi-viewer where you can compare 2 or 4 or 20 images side by side. So in this case, we're looking at trichrome stained mouse lungs, and the mouse on the left has more fibrosis than the mouse on the right, and you can see that by just looking at the overviews next to each other. And actually, one of my favorite tricks in QuPath is using mathematical stain deconvolution. And this is a way of separating out 2 stains that were applied at the same time and separating them so it looks like you're only looking at one at a time. For instance, you could look at just hematoxylin or just eosin in an H&E stain. In this case, where the blue shows just the collagen and the purple shows just the cytoplasm and the nucleus from this trichrome stained lung. And so you can really get a sense of where the collagen fibers are inside this bronchiole. QuPath's annotation tools are really convenient. This is a mouse lung with some tumor metastasis, and we can use the wand tool to very quickly and very easily mark the boundaries of those tumors. This video is playing in real time. It really is that easy to get the contours of the tumors. And then we'll have to go zoom in and get a little one. And then from there, we have a list of the 4 tumors, and we can ask things like, what are each of their areas? Or we can drill down and look at the cells inside each of them. QuPath is a fantastic tool for teaching and collaboration. You can annotate areas, just like I showed you, and then give each object that you mark a name and a description. So in this case, we're looking at an intestine, and we can mark individual goblet cells as goblet cells and talk about how they produce mucus inside the crypt. And it's really easy to share projects with all of these objects attached, either over email or with a thumb drive. I also want to point out an extension to QuPath written by people at the University of Oulu in Finland. This is called QuPath for Education, and it's at openmicroanatomy.github.io. And this is a way for experts to annotate slides and write descriptions and then share them widely with students for free. And it's honestly really neat. And if teaching histology or microanatomy is something you do, or something you'd be interested in trying to do, I highly recommend looking at this extension. There's a demo online that you can play with, and you can see how powerful the tool is. The only problem is the demo is written in Finnish, which I don't speak, but it's a great example of how you can actually do this teaching in whatever language you're most comfortable with. All right, let's get into some deeper analysis. I want to talk to you about a project where we were trying to quantify bacterial infection. We had mice infected with Yersinia, and we took their livers and sectioned and stained them with Wharton starry, which is a silver stain. And our goal was to quantify the number and area of colonies in these livers. From this picture here, the yellow is the healthy liver, all the little circles are the hepatocyte nuclei, and then these brown spots, the larger ones, these are the bacterial colonies that we were trying to measure. To do this, we used a pixel classifier. And the way this works is that you first annotate examples of normal liver tissue versus the bacterial colonies. So in this case, I drew some lines in green over the bacterial colonies and some lines in gray over the normal liver. And then QuPath uses machine learning to take the examples that you just gave it and basically learn the pattern of what these bacterial colonies look like. And from there, it creates this pixel classifier where for every single pixel in the image, it tries to decide if it's part of normal liver or if it's part of a bacterial colony. And then once it has the answer for every picture, it draws outlines around objects. So for instance, you could see it went from this splotch of green to this is one bacterial colony. And this is a process called segmentation, and this is the basis of most QuPath workflows. Once you have these segmented objects, you can count colony numbers or measure areas. For instance, in this case, there were 115 colonies per millimeter squared in this liver, which worked out to 3.5% of the total liver area. You don't just have to segment regions, you can also segment cells. For instance, in this image, this is an immunofluorescence image of a tumor. Every nuclei was stained with DAPI in blue, and then CD8 T cells were stained in yellow, and our goal was to try to count how frequent the CD8 T cells are. So we segmented every cell using the DAPI channel, and the zoom in here shows you approximately what that looks like. And then for each cell, we measured the intensity in the CD8 channel, and then we classified the cells as either positive or negative based on whether they were over a CD8 intensity threshold that we set. And then from there, we can easily calculate the cell frequency. So 4% of the cells in this picture were CD8 positive. And you don't just have to classify cells as positive or negative. You can look at distributions and localizations and all sorts of things. This is a GMSS stained sputum sample, and we segmented the cells just like we did before, and then measured their area. And here I'm showing every cell color-coded by its area. So the yellow cells are the largest ones with areas around 200 microns squared, and the darker colored cells in like the dark purple have a smaller area of around 30 microns squared. And then we can do that for the entire image and get a histogram of the distribution of areas. And then between images, we could compare our distributions. So there's all these tools in QuPath, and when you're working on a project, you're going to want to combine some of the functions to create a custom analysis routine. How you combine this depends entirely on the question you're asking and what you're trying to find, but QuPath gives you the tools to do whatever it is you need to do. So for instance, you might use a thresholder to detect the whole tissue area, then manually annotate just the tumors. Inside the tumors, you might do cell segmentation and cell classification, and then you can measure the distances between cells of different types, or the densities of cells, or whatever it is you're most interested in for your project. And what's really great is the QuPath has this workflow tab where most of the functions that you're doing are automatically recorded. So in this example, I created a full image annotation around the entire square image. And then I used a pixel classifier to find the tumors. I deleted them because I wasn't happy with how it came out. QuPath didn't record as I was doing more annotations to train the classifier better. But then I created annotations again. This time I was happy with it, and then detected cells inside those annotations. And in the workflow tab, you can click on any step here, and it gives you the parameters that you used. So in case of cell detection, it talks about smoothing and minimum sizes and channels. And this is all just recorded for you. And this is a really big deal for record keeping and ensuring consistency, so that if you come back to a project 6 months from now, you'll still have a detailed record of everything you did, so that you can analyze the next image you got in exactly the same way. In addition, you can convert workflows to macro scripts to process a whole folder of images automatically. And so basically for nearly every function in the workflow, there's a way of writing it as a script, and don't worry, there's a button that does that conversion for you. But I promise this is a very, very user-friendly process. It is easy. Even for people who have never scripted before, you can absolutely do this and start batch processing your images and save yourself a lot of time while the computer does the work and you go do something else with your life, like have fun or eat a snack. If you're interested in learning more about how to go from workflows to macros, I highly recommend this paper by Olivia Burry, QuPath, Taking Full Control Through Workflows, Scriptings, and Extensions. And he goes step by step through exactly how you would go about creating a macro. Once you get good at the macros, you can start getting into groovy scripts to perform more advanced analysis that isn't available just through the buttons in QuPath. So in summary, why would you want to use QuPath? It's a bright field and fluorescence image viewer. It's a very user-friendly interface for a lot of common image analysis tasks, finding areas, segmenting cells, doing classification. Honestly, I find myself using it for a lot of small projects, even projects around small images, ones that don't need a pyramid format. QuPath is just so much easier than everything else that it's just my go-to program for everything. But it can handle large stitched images or tissue microarrays or blood smears. It's a great platform for machine learning and deep learning algorithms. I didn't really get into that today, but it has access to the BioImage model zoo and also some models like Stardust and CellPose. It enables macros to automate complex tasks. And it has a lot of tools that enable teaching and collaboration. That being said, there are times when you don't want to use QuPath. There are some projects where other tools are better at the job. For instance, if you're working on something that depends heavily on altering pixel values, you'll want to use something like Fiji. So for instance, if you have to divide one image by another to calculate a ratiometric marker, or you're doing FRET or anything like that, Fiji is a much better way to go. If you're very scared of scripting, but you still want to do batch processing, CellProfiler has a way of batching that is purely on the user interface. You don't have to type anything. Also, if you're doing plate-based high-throughput screening, CellProfiler is more convenient in a lot of ways. It's really set up for that sort of screening. The other big limitation is if you're using 3D images or time series images, I'd recommend Napari or Fiji. QuPath can technically view them, but it is not designed for that sort of analysis. And if you want really deep, detailed control of the absolute latest deep learning algorithms, you probably want to work on Python directly or maybe Napari. Right, so if you are interested in QuPath, where to get help, I recommend the official documentation at qpath.readthedocs.io. This has a lot of explanations of concepts and some good tutorials on the most commonly used functions in QuPath. If you have questions, I really, really highly recommend the ImageSC forum. You can ask anything you want. Beginner questions are great and get, honestly, near immediate feedback from experts around the world. I spend a lot of time there answering questions, and I ask a lot of questions there, and I learn a lot just by reading other people's answers to interesting questions. There's also video tutorials on YouTube at either youtube.com/QuPath or youtube.com/Zbigniew Mikulski. The ones at QuPath are made by Pete Bankhead directly and are really great. The ones on Zbigniew Mikulski's channel are made by either Z or me. And these are step-by-step guides, and they come with example files and focus on some of the more recent additions to the software. All right, I want to say thank you first to Dr. Pete Bankhead for making the software in the first place, and to Zbigniew Mikulski, the director of the core here at the Jolla Institute for Immunology, and also the community at ImageSC for really teaching me quite a lot about image analysis in general and QuPath specifically. And I'd like to thank the Chan Zuckerberg Initiative for my funding.
It's fantastic. That was a really handy presentation. I think I certainly learned a lot about QuPath from watching that. So thank you very much. And I apologize for mispronouncing La Jolla in my rushed introduction to you as we were getting kicked out of our office over here on the ground. I would like to open the floor to, I know that there've been one or two questions already asked and answered in the chat, but we have the rest of the session now essentially to open to anyone who has questions for Sara about QuPath, recommendations for workflows, anything like that. I invite anyone to type in the chat if they don't want to speak directly. Otherwise, feel free to raise your hand and we'll invite you to unmute yourself and ask your question. Emmanuel, go ahead. Are you able to unmute?
Now, yes, thank you. No,
There we go.
it's just not really a question. I just want to thanks Abik to bring a real expert such as Sara McArdle. This is just a thank you, and I just want to take also the opportunity to greet her personally because we had a lot of interaction on the image forum online. So thanks again. Just a thank you, appreciation of them of the talk. Thank you.
Oh, thank you so much for commenting. It's nice to finally meet you.
Anyone else? And that is a fantastic example of how interactive the community can be, and just proof that you can get your questions answered on the forums, such as image.se. And I do encourage everyone to check that out. So in the absence of other questions, Sarah, one of the slides that I actually liked the most out of your presentation was the recommendations of when to use which tools, because I think that's something that can often be very confusing to people, especially if they're new to the field. And there's so many things out there, and better yet, they're all free. They're all open access or open source, right? I wonder if you could sort of offer a little bit of insight, or maybe give an example of where you've seen users maybe realize at some point in their project that it's a good time to switch from one tool to the other, that might help the community think about when they should think about trying a different tool.
I'd say the biggest place where someone realizes, like in the middle of a project, that this tool is not working for them is when they decide to scale up. So they'll have one image and they're happy doing everything by hand and like doing everything manually for this one image, maybe circling every cell or like setting thresholds or whatever it is. And when it's just one or two, it's fine. And then suddenly they do their preliminary study, they had 2 images, everything looked great. They go back to the microscope, they take 10,000 images, and then they try to repeat the same process and go, oh, this is not feasible. So then they suddenly realize they need to like have a tool that scales. So my main recommendation is always do a preliminary experiment, including all the way through image analysis. That is always a good idea. But even once you're happy, just take a moment to think about what the final product, what your final project is going to look like, and if this is still feasible. Yeah.
Thank you. Yes, I that makes perfect sense to me, and that tracks with what I've seen locally. Are you sure that we can get scripts running in QuPath with no coding experience?
Yes.
That seems too good to be true.
Yes, absolutely. For anything that you can do through the user interface, you can absolutely get the scripts running. I've taught people to do it with come in with absolutely nothing. There's no the scripting, the macro language in QuPath is every command reads like the name of the button you would have pressed. So like you would have pressed a button that says cell detection, and then the command is watershed cell detection. So like it's all just like one for one. Every parameter is something you've seen in the user interface. Nothing is new. Nothing is scary. There's a button that goes workflow to macro that gets you most of the way there. They're not usually like perfect. Because the one thing that QuPath doesn't record is like exactly where you clicked. So if you say drew 2 annotations and then clicked on one of them and then wanted to process just the second one, the script has no way of knowing that. So you have to like tell it that. But it's really not hard, and there's a ton of resources online. The official documentation has a good way of getting started. Mike Nelson, who is in the chat, runs a website called imagescientist.com, and he has there a bunch of scripting tutorials, with just like neat little tricks and examples. And then the image forum, we're always happy to help people. Hey, look, he just
there we go. And I suspect I'm going to ask the question that I think Mike Nelson has. Thank you, Mike. Posted a short answer to anyway, but Naomi is asking if it's correct that users can develop their own plugins, and looks like Mike's pointed to a resource. So I wonder if you can comment on that and your experience with that.
Yeah, if you want to make your own plugin, as in something like one expert would develop and then a user would like install, and then they always have the button, it is possible. That one takes a little bit more coding knowledge. You need to know a little bit of Java to make that happen. Not a lot. Again, it's totally possible, and there are guides. But that's just not quite a day one activity. But yeah, if you want to do that, do it. I use user-developed plugins all the time. CellPose is one, and there's a few of them. Image alignment.
Okay, great. Thank you. So there's a question from Wei Chen about file formats. What file formats are used in QuPath, and if big stitched images or folders can be used?
QuPath prefers big stitched images. Sorry, I assume you mean by a folder, you mean one that is going to be stitched. Basically the tiles from a large image. You should stitch them together before working in QuPath. But if you just have like individual fields of view and you just want to batch process 1,000 images in folder, it can handle that. Like it just reads them as individual images and processes them all.
Okay, great. And it would take, I assume, a .tiff file format or any of the commercial file formats?
Yeah, I missed that part of the question. Yeah, TIFF, OME TIFF, and then anything that either bioformats or OpenSide can read. So that's everything by Leica, Zeiss, Nikon. You can read all of them. The only thing that it really struggles with is certain scanners. The 3D Histotext scanner produces a .mrxs file, which is not bioformats compatible. And so it can read, I believe it can read bright field, but not fluorescence. The 3D Histotext scanner files are weird.
Okay.
Yeah.
But most common file formats, it can handle fairly well.
Yeah. And anything you can put into a TIFF, it works.
Brilliant. So there's a question asking for confirmation from Adijoka, asking that it sounds like we need coding to use QuPath. I'm going to invite you to dispel that.
No, absolutely not. You can do everything just through point and click. There's a user interface. And you could just like, you never need to actually code anything. If you are comfortable coding, you can make everything faster and save yourself some time, and that's pretty great. But if you don't want to do that, basically everything you need would be available to you through just the point and click interface.
Great, thank you. So I think that's all the questions that have come up so far in the chat. I'm going to invite, we picked a little bit of momentum there, so I'm going to encourage anyone who does have any questions, do feel free to type them or just raise your hand. Emmanuel was brave enough to raise his hand. More of you are welcome to do it as well. I'm going to give everyone one more minute to see if they've got burning questions. In the interim, Sarah, you pointed to a number of really good resources in your final slides, and I think you and Mike have both posted links to them. Would you say those are sort of the future tutorials, and the QuPath documentation is the best places for people to get started with QuPath?
Yes, I'd recommend. I'd say the place to get started, if you prefer reading your instructions, is the documentation. If you prefer watching your instructions, is Pete's video tutorials. And then from there, me and Mike and a bunch of other users have all like made different tutorials around different aspects of the program. But Pete's are like the really intro stuff that you need like immediately. He's also just a very good speaker and is just very clear and slow. I know I tend to talk fast.
You're not the only one. I'm guilty of that too. So I've long been a a fan of Pete's work, including his just bioimage analysis training. So that's fantastic.
Yeah.
In the absence of more questions, Sarah, I'm going to put you on the spot and ask you if there was one thing you wanted people to know about QuPath and take away from this, if you could sum up in like a sentence or two. And then in the absence of questions, I think we can call it there.
One thing to know about QuPath. Honestly, I guess my like one takeaway is that it's worth just like trying it and just trying different things in it. Basically, you're never going to break your image by pressing buttons in QuPath. Like, there's nothing you can do that will permanently damage your files, your computer, nothing. So like, play around with it, see what happens. See, every function comes with some default parameters. See what happens when you change them. See what happens when you do things in different orders. Like, just like test some stuff, and you end up learning a lot about your program, about image analysis in general, about your images. Like, sometimes something you didn't think would work really, really does, and you get something really cool out of it. So just like, go at it, see what you can find.
I actually really love that. It's a reminder that the analysis is as much part of the experiment as the rest of the lab work.
Yeah.
That's really great. Sarah, thank you so much for your time and being willing to share your experience and expertise. I'm not seeing any more questions, so I think you've probably done. I think you've done an amazing job of covering and giving an overview of QuPath. Thank you to Mike and others posting links and other assistance in the chat. Thanks once again to Precious for coordinating this and making this happen today. And thank you to everyone for joining.